Good morning students, I am Dr. Sonal Gupta and I am working as an assistant professor at K.L. Mehta Dayanand College, Faridabad, and that is affiliated to MD University Rohtak. Now the module is the clinical trials of biopharmaceuticals, and the learning objectives of this module are: number 1, what are biopharmaceuticals and its different categories; number 2, the 4 clinical phases of this biopharmaceuticals; and number 3, the challenges involved for small scale biopharmaceutical industries. Now the introduction. What are biopharmaceuticals? Biopharmaceuticals are medical drugs produced using biotechnology. These are basically nucleic acids, DNA, RNA, antisense oligonucleotides, proteins that could be monoclonal antibodies which can be used for therapeutic or diagnostic purposes. These are basically prepared from originally non-engineered biological sources. Biopharmaceutical drugs structurally mimics compounds found within the body and are produced by applying different techniques involved in biotechnology. These have the potential to cure diseases rather than merely treating symptoms, and have fewer side effects because of their specificity. Further describing about the biopharmaceuticals, we can explain it like the large majority of biopharmaceutical products are pharmaceuticals that are derived from life forms. Small molecule drugs are not typically regarded as biopharmaceuticals in nature by the industry. However, the members of the press and the business and financial community often extend the definition to include pharmaceuticals not created through biotechnology. That is, the term has become a buzzword for a variety of different companies producing new, apparently high technology pharmaceutical products. When a biopharmaceutical is developed, the company will typically apply for a patent, which is a grant for exclusive manufacturing rights. This is the primary means by which the developer of the drug can recover the investment cost for development of the pharmaceutical. The patent laws in the US and Europe differ somewhat on the requirements for a patent, with the European requirements are perceived as more difficult to satisfy. The total number of patents granted for biopharmaceuticals has risen significantly since the 1970s. In 1978, the total patent grant was 30. This had climbed up to 15,600 in 1995, and by 2001 there were 34,527 patent applications. As we have already explained, the biopharmaceuticals are drugs which are produced by the means of biotechnology. By this, we want to say the number of ways in which such drugs can be made, but the key distinction between them and the other drug is that they are not extracted from a native source or synthesized with chemical reactions. Instead, they are created with the use of living organisms which may have been modified to produce the desired compound. This requires the use of specific equipment and clean rooms for safety, which protects the integrity of the pharmaceutical compounds while they are produced and packaged. One of the classic methods of making biopharmaceutical products involves the use of a bioreactor. A bioreactor is a container which is used to create tightly controlled conditions which facilitate the growth of a particular organism. In a bioreactor, drugs can be produced by organisms which generate biopharmaceuticals as a byproduct of their life cycle, often because these organisms have modified to produce specific proteins and nucleic acids. Cell cultures and modified microbes can be both used in bioreactors to make drugs and compounds which can be used in the production of biopharmaceuticals. A variety of substances can be made using biopharmaceutical technology, including blood factors, interferons, hormones, vaccines, and monoclonal antibodies. The first biopharmaceutical product to hit the market was artificial human insulin, also called as Humulin, that was released in 1982. This product was first developed by Genentech company and it was marketed and manufactured by Eli Lilly. The biopharmaceuticals industry exploded after this product. As we have explained, the first product in this category was human insulin, also called as humulin. The cloning of human genetic material and development of in vitro biological production systems has allowed the production of virtually any recombinant DNA based biological substance for eventual development of a drug. Monoclonal antibody technology combined with recombinant DNA technology has paved the way for tailor-made and targeted medicines. Gene and cell-based therapies are emerging as new approaches. Recombinant therapeutic proteins are of a complex nature, which are composed of a long chain of amino acids, modified amino acids, derivatized by sugar moieties, folded by complex mechanisms. These proteins are made in living cells that could be a bacteria, yeast cell, animal or human cell lines. The ultimate characteristics of a drug containing a recombinant therapeutic protein are to a large part determined by the process through which they are produced. Choice of cell type, development of the genetically modified cell for production, the production process, the purification process and the formulation of the therapeutic protein into a drug. Now we will cover the second objective, that is how biopharmaceuticals are different from chemical based drugs. Compared with other types of biopharmaceutical products, these products are derived from a biological source or a biotechnological process, which are structurally complex and involve manufacturing processes that require tight control to ensure their safety, quality and efficacy. Biological products, because of their sheer size, are orders of magnitude more complicated than small molecule drugs. This can be seen by comparison of molecular weight, which can be used as a measure of the size of a given product. Moreover, the product arising from a manufacturing process is often not a pure homogeneous mixture. Rather, various forms of these molecules are usually present in the final product. The next point covered to be the difference between the biopharmaceutical products and the conventional small molecule chemical drugs. In this category, the chemically synthesized products are also known as small molecule drugs, since as an example we can quote the aspirin. The molecular weight of aspirin is 180 Dalton. However, in case of biopharmaceuticals, these are complex macromolecules. For example, interferon is a molecule that is weighing around 19,000 Daltons with complex structural and appropriate biological activity requirements. Biopharmaceuticals have more potential heterogeneity than small molecule drugs. The large majority of pharmaceutical products are derived from life forms. Small molecule drugs are not typically regarded as biopharmaceuticals in nature by the industry. The nature of the manufacturing process and the safety and efficacy profile of biopharmaceutical products are also different. The majority of first generation biopharmaceuticals are unengineered murine monoclonal antibodies or simple replacement proteins displaying an identical amino acid sequence to the native human protein. Modern biopharmaceuticals are engineered second generation products. Engineering can entail alteration of amino acid sequence, glycol component of a glycosylated protein or the covalent attachment of chemical moieties such as polyethylene glycol. Engineering has been applied to alter immunological or pharmacological profile of the proteins or to generate novel fusion products. The next objective of the module was to cover the 4 different phases of clinical trials of biopharmaceuticals. A clinical trial is an experiment testing a medical treatment on human subjects. The participation of humans means that we need to execute every aspect of clinical research to the highest ethical standards, and the protection of participants' welfare is our paramount concern. Regulatory agencies worldwide govern how these trials are to be conducted. For example, the agencies like the FDA, the European Medical Agency and the Japanese Pharmaceuticals and Medical Devices Agency. Now, when we understand what are clinical trials of biopharmaceuticals, we need to know the International Conference on Harmonization of Technical Requirements for Registration of Pharmaceuticals for Human Use, that is ICH, brings together regulatory agencies and biopharmaceutical industry representatives from Europe, Japan and US to discuss scientific and technical aspects of drug registration, that is the marketing approval. Its mission is to achieve greater harmonization to ensure that safe, effective and high quality medicines are developed and registered in the most resource efficient manner. Biopharmaceutical clinical trials are typically classified into various phases, with any given trial being identified as belonging to one of them. A common system includes 4 phases, phase 1, 2, 3 and 4. However, as ICH guidelines, it is important to recognize that the phase of development provides an inadequate basis for classification of clinical trials because one type of trial may occur in several phases. The guidelines provide an alternate 4-item system, one that employs more informative nomenclature related to study objectives. This system includes human pharmacology, therapeutic exploratory, therapeutic confirmatory and therapeutic use trials. Guideline E8 hence combines these systems as indicated by the headings of the next 4 sections. The phase 1, which is also called as human pharmacology. An enormous amount of work spanning several years is completed in the new drug development process before clinical trials commence. This work comprises a drug's non-clinical development program. Non-clinical development comprises in silico, in vitro, ex vivo and in vivo testing, including investigations conducted at the intracellular, cellular, isolated tissue, isolated organ and intact animal levels. No animal model is a perfect predictor of the precise effects of a drug in humans, but these data nonetheless provide the rational basis for determining what drug doses we administer to the individual participants in first-in-human clinical trials. Phase 2, that is also called as therapeutic exploratory phase. Participants in these trials have the disease or condition of clinical concern, example, hypertension, thus facilitating initial assessment of a drug's safety and efficacy in the intended patient population. They are conducted by researchers trained in clinical trial methodology and operational execution. Often, several hundred participants take part in these trials. Some of them receive the drug being developed and some receive a control treatment, which can be a placebo or an active comparator, that is existing product in the nature. The nature of these trials is therefore comparative, since responses to the drug are compared with responses to the control treatment to investigate the drug's comparative efficacy. Phase 3, that is also called as therapeutic confirmatory phase. Therapeutic confirmatory studies are conducted as RCTs. These are also called randomized clinical trials. Several thousand participants with the disease or clinical condition for which the drug is being developed takes part. These trials are often required to specifically include certain subgroups of participants that are representative of patients who will receive them in clinical practice if the drug is approved. And the last phase is phase 4, that is also called as therapeutic use. Therapeutic use studies are conducted once the drug is on the market. They may be optional studies or studies required by a regulatory agency as a condition of approving the drug for marketing. This phase actually explores a drug's place in current treatment practice guidelines, comparing its safety and or efficacy with other treatment options or combination of various treatment options. In the latter case, the regulatory agency approving the drug for marketing felt that based on the drug's benefit-risk balance as indicated by the data, they had to review at that time the drug could be marketed and hence offer immediate benefit to the patients. Now, the different categories of these biopharmaceuticals. Number 1 is hematopoietic growth factors. Now, the next portion is current practices in the clinical trials of biopharmaceuticals. Biopharmaceuticals are being developed to fight cancer, viral infections, diabetes, hepatitis, and multiple sclerosis, and these can be grouped into various categories. For example, hematopoietic growth factors. The hematopoietic growth factors are glycoproteins that stimulate the proliferation and development of clonogenic precursor cell populations. Through recombinant technology, hematopoietic growth factors administered at pharmacological doses can provide significant clinical benefit for the cancer patient undergoing chemotherapy, as hematopoietic growth factors serve to stimulate the production of mature blood cells. Their clinical application in the diseases characterized by suboptimal production of specific blood cell types was obvious. As one of an example, we can quote recombinant human erythropoietin was purified and cloned in 1977 and then commercially produced as epoetin alpha 3. It was initially approved in 1987 as a treatment for anemia in chronic renal failure patients on dialysis and in 1990 as a treatment for patients with HIV-related anemia. In 1993, the indication was expanded to include chemotherapy-related anemia. The approval was based on a randomized placebo-controlled trial with 3 subsets including anemia cancer, patients who receive platinum-based chemotherapy, non-platinum-based chemotherapy or no chemotherapy. The non-chemotherapy patients received a lower dose of epoetin alpha for a shorter period and the hemoglobin improvement did not reach statistical significance. The chemotherapy groups received 150 units per kg epoetin alpha for 12 weeks, resulting in an improvement in hematocrit of approximately 6 points in cisplatin and non-cisplatin groups compared with placebo. In addition, a statistically significant reduction was seen in transfusion requirements and overall QOL in the epoetin alpha 3 group compared with the placebo. Adverse events were similar in the 2 groups and largely reflected the underlying disease and chemotherapy. Now, this table gives us some other examples of hematopoietic factors that are in medicinal use or in clinical trials. The second category is cytokines. Cytokines are hormone-like molecules that can control reactions between the cells. They activate cells of the immune system such as lymphocytes and macrophages. Interferon is a potent glycoprotein cytokine that acts against viruses and uncontrolled cell proliferation. Interleukins functions as messenger for various steps in the immune process. Several interleukins, particularly those capable of modulating transformed cell growth as well as those exhibiting immune stimulatory properties, enjoy significant clinical interest. As with other cytokines, the advent of recombinant DNA technology facilitates the production of these molecules in quantities sufficient to meet actual potential medical needs. Now, this next category was cytokines. The first interleukin to be approved for medical use was interleukin 2 that was approved in 1992 by the FDA for the treatment of renal cell carcinoma. Several other additional interleukin preparations are currently in clinical trials, as we can observe by this table. The third category is enzymes. These are complex proteins that cause a specific chemical change in other substances without being changed themselves. And this table shows the thrombolytic agents or enzymes approved for the general medical use. The next product in this category are vaccines. These are microorganisms or subunit of microorganisms that can be used to stimulate resistance in a human to specific diseases as well as to stimulate immune responses. For example, hepatitis B virus, Ebola virus, etc. Researchers have identified a protein in infected liver cells that is essential for hepatitis C virus replication. Inhibiting this protein is highly efficient in blocking the virus replication. Now the next category is antisense therapy. One of the treatments for genetic disorder or infections is antisense therapy. When the genetic sequence of a gene is known to be causative of a disease, antisense drugs hybridize with the inactive form of mRNA, thereby restricting a particular gene from producing the protein for which it holds the recipe. Although specificity and selectivity are the key features of antisense oligonucleotides, the need to target the right tissues and reach the nucleus remains a challenge that is yet to overcome. The product is peptide therapeutics. Peptide therapeutics characterizes a novel class of therapeutic agents. Currently, only one selected cationic antimicrobial peptide has been licensed, that is for topical application. It is now possible to produce very large quantities of therapeutic peptides with tight specifications by using the wide possibilities offered by liquid phase and solid phase technologies, alone or in combination depending on the specific features of a given project. Moreover, many peptides currently in the preclinical or clinical stages contain non-natural amino acids that could be beta amino acids or amino acids having D configuration to make them more active or more stable. Selection methodologies addressing protease resistance have been developed, and when combined with methods such as pegylation antibody Fc attachment and binding to serum albumin look like to finally turn therapeutic peptides into a widely accepted drug class. The last product is cell therapies. Cell therapy designates the process of introducing new cells into tissues in order to treat a disease. Several stem cell therapies are routinely used to treat disease today. Adult stem cell transplants, example, bone marrow stem cells, peripheral blood stem cells and umbilical cord blood stem cell transplant. Umbilical cord blood stem cell transplants are less prone to rejection than the other bone marrow or peripheral blood stem cells. The best known stem cell therapy to date is the bone marrow transplant, which is used to treat leukemia and other types of cancers as well as various blood related disorders. Stem cell based therapies. The big challenge for the stem cell community is therefore to facilitate the best possible interaction with the population at large, that is, one stem cell world. Stem cell treatments are a type of genetic medicine that introduces new cells into damaged tissues to treat the disease or injury. Many medical researchers believe that stem cell treatments have the potential to change the face of human diseases and alleviate the sufferings. The ability of stem cells to self-renew and give rise to subsequent generations that can differentiate offer a large potential to culture tissues that can replace disease and damaged tissues in the body without the risk of rejection and side effects. A number of stem cell treatments exist, although most are still experimental or are costly, with the notable exception of bone marrow transplantation. Medical researchers anticipate one day being able to use these technologies derived from adult and embryonic stem cell research to treat cancer, type 1 diabetes mellitus, Parkinson's disease, Huntington's disease, celiac disease, cardiac failure, muscle damage and neurological disorders, along with many others. In conclusion, we can explain about the unique challenges of clinical development by small companies that are often addressed with this use of smaller clinical development programs than those used by large companies. Development of drugs for rare diseases may be used as a strategy, but the small size of the populations with such diseases and the small samples available for trials require approaches that can maximize the power to detect efficacy, which can include the use of historical controls, new surrogate endpoints, or enrichment for participants who are likely to have a response. Temptations to use uncontrolled early small studies to support further development of products may prove to be problematic. Therefore, small companies with limited resources require both innovative approaches and the rigor for success. Here, this table illustrates some examples of approaches used by small companies that achieved the FDA approval for their drugs in 2014 and 15. In the end, these small companies, however, could use a variety of approaches to address the above challenges, including the use of new technical platforms, the use of new formulations or technologies that enhance the actions of known drugs, or the use of trial designs that take advantage of the specific market they hope to enter. Other companies develop products that are spun off from or licensed from large companies. In fact, many small companies may choose to partner with larger companies to add resources and experience. Many small companies can also repurpose drugs or pursue narrow niche markets such as rare inherited diseases, uncommon cancers, or specific infectious diseases to remain viable. Thank you.